Hello, everyone, and thank you for joining. We'll give everybody a few more minutes to join the webinar. We're going to get started at 10 a.m. Eastern, so in about 3 minutes. Hello, everyone. I want to thank everybody for attending today's webinar. Just before we get started here, if you have any questions during the webinar, please enter them in the chat window. We will do our best to answer them as we go. We'll also have a Q&A session at the end of the webinar to address any other questions that we may have at that point. A quick introduction about myself. My name is Pavel Sul. I am the Chief Technology Officer here at MIPAR. I've been with MIPAR for about 5 years now, and before then I did biomedical research at The Ohio State University. So without further ado, I'm excited to announce MyPart version 4.1.0, the update, which has really been focused on the high throughput analysis in life science and primarily biomedical research, high content screening. The agenda for today's webinar is fairly straightforward. We're going to try to do this fairly efficiently, just to be able to highlight the features and answer any questions that you may have. So we're going to introduce MyPart for those who are new to the software. We'll then jump into a hot content screening analysis demonstration. We'll then review a new project to full automation best practice approach. Then we will talk about some features that are in development currently. We'll then highlight MyPart Training offerings, and then we'll have some closing slides. For a quick introduction of MyPart, it was developed at the Ohio State University Department of Material Science. Even though it was developed for material science, we quickly found out that the interface was ideal for developing and running computer vision algorithms. So since then, We have worked in life science, satellite imaging, electron microscopy, of course, and have had quite a bit of success working in a wide range of applications, which we believe is due to the versatility of the software. MyPart was established in 2017 in Columbus, Ohio. We're still in Columbus, Ohio. MyPart was originally developed by users of the software, and to this day is still being developed by people who use it every single day. This ensures the highest quality of software, as well as a user-friendly interface. We like to say if we have a hard time using the software, chances are our customers will as well. So we put user experience at the forefront in what we do. This mindset has let us work with hundreds of universities, research institutes, and companies worldwide. I think we have customers in over 40 countries at this point, and we have regional distributors in Europe and Asia. So the demonstration that I will be covering today will highlight some of the new features in version 4.1.0. These features were developed in collaboration with our customers who work in this industry, and we're able to incorporate really the nuance of what they have to deal with day to day into the workflows and feature set of this release to help improve the overall experience of MyPart when working with these high throughput applications, which generate gigabytes and gigabytes of data. Primarily, the challenges come from handling multi-channel information, and we'll highlight how what improvements we made in both the image processor and the batch processor, so single image processing as well as batch processing of such data sets. We then improved our ability to handle intensity measurements in higher bit depth than just 8-bit. This was really a requirement because most high content screening applications operate under higher bit depth. This is really used to separate or improve the sensitivity of the assay. And lastly, we revamped our channel operation recipe step to be able to handle more complex operations and to maintain floating point precision until the final output. We also added some new settings for the output of the image to help improve the accuracy and the power of the recipe step. To review the sample application, this specific data set is 4-channel data. It's collected using a high content screening system. This application, this data set was collected to study the stimulation inhibition of translocation process of NF-kappa beta from cytosol to nucleus. And the detection challenges require us to find the nucleus, cytosol, nucleus boundary, and the complete cytosol. So without further ado, let's go ahead and set up my screen share. Okay. So those of you that are familiar with MyPart, you'll find this interface, really the home base of the software. Those of you that are new, this is what we call the image processor. This allows us to open images and apply what we call a recipe or an algorithm to the image in order to do a detection and a measurement. So let's go ahead and do that. So the data set that we're working with is a classic data set where we have that comes off of a high content system where we are collecting a well, a site in a well, and then a wavelength, and then sometimes you'll have a Z. But in this case, we have 4 channels per site, and then we have multiple sites per well. When developing algorithms, you really want to optimize for the wavelengths here. So let's go ahead and select the 4 wavelengths we would like to work on. MyPart automatically stacks the image into a multi-page TIFF. You're then able to access individual channels right from multi-stack using the reference view here on the left, as well as our channel operation tool. So a quick recipe that we have put together around this application to highlight the detection. Let's go ahead and select this recipe. So I have that recipe loaded in our recents menu. When you're using MyPart and you're working with the recipe a lot, it will be added to this recents menu, allowing you to select it from a quick drop down. What you see here is the application of a artificial intelligence-based recipe in real time. I'm working on a professional grade laptop. None of this is being sent to the cloud. This is done locally on my system. When the recipe has been applied, MyPart automatically outputs a report summary, as well as it overlays the detection over the original image. Let's change this back to color, maybe make it a little bit easier to see. So what we really have here on the right is the detection of both the nuclei and the cytosol. And the thing that I want to highlight is that all the detection has really been done by the deep learning recipe for us. So, or the deep learning model. In this case, the nuclei have been highlighted. Anybody who has very little to no experience with image analysis will be able to very easily pick up the nuclei from this image, right? That's kind of the power of deep learning. It's able to simplify fairly complex solutions for us. But relative to some other problems in life science, being able to find the nuclei is very straightforward. The thing that we're much more excited about is being able to highlight boundaries like this, right? So we're able to find the boundaries between cells with a high degree of accuracy. And again, this significantly simplifies the problem for anybody who's looking to quickly develop an algorithm to do this type of analysis. So to demonstrate a full transition of from a single site to a full column of data, let's go ahead and run this, or demonstrate how we would run this analysis on a full data set. So let's go ahead and launch our batch processor. If we launch it from the image processor, it will automatically be opened up with a recipe here on the left. I can go ahead and add my entire data set by selecting all of the images in the folder. I want to specify how many channel data I'm working with. In this case, that's 4 channels. I do want to output the feature measurements. And let's go ahead and set a save location. We'll do our batch folder. And let's give it a session name. Let's go ahead and call this complete. So the session name is important. It just allows you to go back and review a session in case, for whatever reason, you need to pull up the data again in the future. Once this is set up, I just click process, and MyPart goes on and it does what it does best, and it does our detection and measurement for us. So we'll let it get through a single set here. But as to not to take up too much of everybody's time, and through the magic of being able to do this ahead of time, I will stop this batch prematurely, and we'll review the session together. So we'll see we're able to process this data set in about 22 minutes, and this is again this is happening in real time on my system, and I'm also doing some screen sharing. So let's go ahead and stop this session for now. Let's pull up our session processor, load in our pre-made batch so we can review the results together. Right, so what we're able to do is see exactly what was detected. We can jump from site to site. We can jump to other wells. And the wells in this case, as in most high content screening, is just a concentration gradient of a reagent. So as we move down through the wells, the concentration gets lower. But you can see that we're able to reliably detect the nuclei in the complete cytosol in every single site. So the question really becomes, how does MyPart handle the data output? What we can see is that if we go to the location where we ran our batch, we go to our measurements, it breaks up our results into 2 folders. Each folder will contain the site, the cytosol, and then the feature measurements that are included in there, which is our centroid position of the cytosol, as well as our intensity mean in 16-bit. This data is always saved as CSV. You can then pipe this data into your favorite statistical analysis software and do the same type of analysis you normally do in these types of problems, where you plot your concentration gradient versus an intensity ratio or intensity difference. Or you can use more powerful solutions, be able to take this data and transfer it into AI-based data mining, data analysis solutions, where they may be able to find some insight in the data outside of just the classical applications. But what I'd like to do today is really talk about MyPart's ability to quickly make robust recipes. So I have this recipe, right, and I'm able to find the nuclei in the cytosol, but let's say that I have a hypothesis that my marker of interest is not necessarily diffused entirely through the cell cytosol, but it's localized around the nucleus. So what I can do is very quickly say, okay, well, it's because it's around the nucleus. Let's go ahead and disable these steps. I'm going to go ahead and select my nuclei. I'm going to set them as a companion image. I'm going to dilate my nuclei 3 points. I'm going to subtract my original nuclei, and then I have this awesome ability to just look at the region outside of the nucleus. Let's set this as memory image 3. Go ahead and remove all of these extra steps and re-enable my measurement setup. Right, so let's go ahead and switch this back over, and now we can see that I'm no longer looking at the complete cytosol. I'm looking at the localized region around the nucleus. Let's go ahead and regenerate my report. It's still in 16-bit. And I can say, okay, let's save this recipe, boundary, and launch the batch processor one more time. Our data is still in here. I'm combining my channels. I'm outputting my feature measurements. Let's go ahead and this is where we want to be saving our batch. Let's give it a more meaningful name. Let's just say boundary. So webinar, and then we will again click process. And in a matter of, I don't know, a minute, I was able to entirely change the region of interest where I'm looking at my intensities and be able to explore a completely different hypothesis. relying that my detection is going to be accurate, thanks to the powerful tools that the MIPAR has to offer. So once again, I'm going to go ahead and cancel this batch. Let's go back to our session processor and reload our boundary data set. And what we can see is that just as before, we're able to jump from site to site, from well to well, and we know that our detection is accurate. And just as before, we can take a look at our measurements. Again, they are in a friendly format. You can take this data and pipe it into statistical analysis software or downstream AI-based data analysis solutions to do the data analysis. So let's go back to the image processor one more time, and I would like to highlight some of the new features in our channel operation tool. Now, this may not be applicable to this specific assay, but let's say that I'm not interested in generating my intensity measurement relative to just a single channel, but let's say I have some markers in channel 3, and I want to find the difference between those two channels, and I want to divide that by the original channel to give me a ratio. So what you're able to do is very quickly type in an equation between those channels. MyPart will maintain the floating point precision during that arithmetic and output the measurement again in our original 16-bit bit depth to maintain our higher level of sensitivity. I can then go ahead one more time, save a recipe as channel ratio, and I can once again launch the batch processor and go through this process. Now, in about 5 minutes, I was able to demonstrate how we can explore 3 separate hypotheses when it comes to this type of analysis without really having to worry about the image analysis aspect of this. So really what the power that MyPart brings to your laboratory is being able to have the freedom to explore hypotheses without having to worry about whether or not your image analysis can do it. You're also free to explore putting together more complex assays, right? You're no longer bound to hoping that the out-of-the-box assay that you receive will come with some image analysis solution, or hoping that somebody else has created the solution for you. With MyPart, we're able to readily put these solutions together in a matter of minutes and execute them on your entire data set to explore your hypothesis. So let's go ahead and jump back into our slide presentation. Okay. So that was a fairly quick demonstration of some of the features that we've included in the high throughput analysis update of MyPart 4.1.0. Now, if you're new to MyPart, the question always becomes, well, how do I get these recipes? And we put together a diagram here to help new users navigate this process. And it really starts with whether or not you have annotated data, and it doesn't have to be much. We've been able to put together robust deep learning-based solutions with as little as 10 images that have been able to process hundreds, if not thousands, of images downstream. If you do not have any annotated data, that's okay. Again, we can start out with something called a starter recipe, which uses more conventional image processing tools to do the detection for us. Now, what we recommend is, most new users jump in and they work with an application specialist to put together these recipes. But as soon as that's complete, they're able to apply this recipe to their data, continue to get the results to do their day-to-day work, which those of you that work in research know that really time is of the essence here. So being able to continually produce the measurement data that you need is critical, because you don't really want to stop your day-to-day operations in order to create image analysis. But what this allows you to do is then take the results from your batch and retrain your data set. This improves the robustness of your artificial intelligence recipe. Or if you started with a starter recipe, you're able to take those results and create an artificial intelligence-based recipes, which just makes it more robust, actually simplifies the solution in the long run. But like I said before, our application specialists, which hold a PhD education in their respective field, are here to help you with your project. They will bring the experience they have from their specific field, combined with the image analysis experience they have at MIPAR, to help you put together a robust workflow. So let's go ahead and move forward to some exciting announcements. A couple of things that I would like to cover is that we've been working on developing a MyPart capture extension. Those of you that are working with optical data, we'll have a full list of camera support coming soon. But we're excited about this feature because it brings the full recipe automation to optical image analysis, and being able to control the capture is a huge part of what we do. Being able to control the capture aspect of the image analysis process allows us to have further control over the imaging conditions and better improve the workflow. We're also excited to announce that we're working on advanced feature measurements, and this allows users to customize measurements using our existing library of measurements, which is, I think, over 30 measurements now. And then you can, of course, assign customized names to those measurements to make sure that it's still readable and user-friendly to others who you may be sharing that application with. The other thing I would like to talk about is MyPart Training. So we are offering training virtually via GoToMeeting or Microsoft Teams. We offer this training globally. We also offer in-person training here at headquarters in Columbus, Ohio. If you're interested in that, please reach out to support@mypart.us for pricing and scheduling. So I would like to now pause and see if there's any questions that we may have. If you have any questions at this point, feel free to type them into the chat window. If not, or if you think of something afterwards, feel free to email us at support@mypart.us or sales@mypart.us, and we'll be happy to help you out. You'll also receive a replay of this webinar via email. So feel free to share that with a colleague who may have found this webinar interesting. And again, don't hesitate to reach out if you have any questions. We'll give it a couple of minutes in case somebody has any questions, and then we will move to the closing slides. Let's see. It looks like you should have been able to download a MyPart in your lab document, which highlights some of the benefits of using MyPart in a life science laboratory. There's a lot of good information there in case you're interested. Okay, if there are no other questions, let's do a quick closing slide. If you'd like to learn more about MyPart, feel free to reach out at sales@mypart.us or call the number that's on your screen. You can also submit an image that you're working on following that link there. Again, you will receive all this information in the follow-up email. Also, if you like, you can follow us on LinkedIn or Twitter. We post a lot of interesting projects that we're working on those platforms. Okay, well, I thank everybody for taking the time to attend our quick webinar. We hope to have more of these in the near future as we continue to develop more and more features and highlight how these are able to support different applications around the world. Thank you for your time, and have a good rest of your week.